Hi, my name is Jana Runte and I am a PhD student in the Together Unique study, which is a collaboration of the University of Leiden and the Vrije Universiteit Amsterdam. I investigated the heritability of the association between sleep and cortisol. Past research indicated that sleep is associated with cortisol. Lower sleep quality seems to be related to higher morning cortisol levels. However, many studies use self-reports as indicators of sleep instead of objective measures. Also, a lot of studies looked at insomnia or sleep problems and not at sleep in a healthy population. And most sleep studies were conducted on adults or adolescents, and when they were conducted on children, sample sizes tended to be small. Furthermore, genetic factors seem to play a role in sleep and cortisol. Twin studies suggest heritability indices between 30 and 60% for both sleep quality and cortisol levels. However, behavioral genetic studies either investigated sleep or cortisol, but not both factors in relation to each other, although sleep and cortisol have consistently been found to be related. In our study, we measured sleep over 4 consecutive days using actigraphs. Sleep duration, sleep efficiency, and wake episodes were used as indicators of sleep quality. Cortisol was measured 3 times a day over 2 consecutive days. Basal morning cortisol levels and the area under the curve with respect to the ground, which is the total cortisol production during the day, were used in the analyses. In our study, we investigated the association between sleep and cortisol in a classical twin design. We expected that genetic factors significantly explain variance in sleep duration, sleep efficiency, and wake episodes. Also, genetic factors significantly explain variance in basal morning cortisol levels and the area under the curve. Third, we expected that a common genetic factor significantly explains the covariance between the sleep measures and the cortisol measures. Because previous research has found genetic influences on cortisol and sleep, and both are known to play a role in the circadian rhythm, which makes it likely that the same genetic influences explain a part of the covariance in sleep and cortisol. And lastly, we expected that shared environmental factors significantly explain variance in sleep duration, sleep efficiency, and wake episodes. Previous studies did not find shared environmental influences. However, the samples in previous studies were not children. We expect parental rules about sleep times and school times to be possible influences on sleep indicators such as sleep duration. Our study is a longitudinal twin study consisting of 6 yearly visits with additional ambulatory assessments. We used data collected between 2015 and 2019. We had a total of 436 twin pairs with a mean age of 7.5, and around half of the twins were boys. You can see that the correlations of the monozygotic twins are always, for all variables, higher than the dizygotic correlations. This speaks for a genetic factor. We tested this afterwards. We examined the relative influence of genetic and environmental factors using behavioral genetic analyses. We tested to what extent variance in a trait can be explained by either genes, shared environmental factors, or unique environmental factors. Monozygotic twins share 100% of the genetic makeup. Dizygotic twins share 50%. If sleep and/or cortisol levels in the monozygotic twins are significantly more similar to each other compared to the similarity in dizygotic twins, this is an indication of genetic effects. But first, we ran a common pathway model to look whether there's one common factor that is influenced by A, C, and E that accounts for variations in the sleep variables. But this was not the case because the model fit poorly. We then tested our hypothesis with this model. The data fit the model we ran on sleep duration and basal morning cortisol levels. We could drop the C component without significantly worsening the fit of the model, or we could not drop the A component. What we can see is that genetic factors influence variations in sleep duration, as well as variations in basal morning cortisol levels. Also, unique individual factors influence variations in sleep duration and in basal morning cortisol levels. But it seems to be that not the same genetic factor influences both sleep duration and basal morning cortisol, which can be seen at the low path coefficients. We found the same pattern for the models on sleep efficiency and wake episodes, significant A and E, but not C, and no common A and E for both. We ran the same analyses with area under the curve instead of basal morning cortisol levels and the 3 sleep variables, and we also find the exact same results. What we can conclude from this study is that we can confirm that also in children, it seems that variations in sleep duration, efficiency, and wake episodes are to a certain extent heritable. Also, variations in the phenotypes, cortisol and levels and the area under the curve are to a certain extent heritable. And although it seemed quite logical that sleep and cortisol might be affected by the same genetic factor, this doesn't seem to be the case. No common genetic factor underlies these phenotypes. And interestingly, despite parental rules on sleep and wake times, shared environmental factors did not explain variations in the sleep measures. For exploratory reasons, we looked at whether this is only the case in twins that sleep in separate rooms, but our exploratory analysis on children with a shared bedroom did yield the same results. Now I would like to thank the ELSA team, my supervisors, and the University of Trier. Thank you.